current climate change experimental toolbox. So I'll talk about some of the range of techniques that researchers have at their disposal, and I will talk about some of the strengths and weaknesses of those different techniques. And what I hope to really convey is that no one technique is really going to give us all the answers, and rather it's by combining a combination of different techniques at different scales and using different methods that will get a much deeper understanding about climate change impacts on coral reefs and all marine ecosystems. And then to give you a coral reef example, I'm going to talk about my work where I've tried to use multiple experimental approaches to study how net reef calcification will be affected in a high CO2 future. And then at the end of my talk, I'll talk a little bit about some future approaches that we're trying to develop in my research group, and hopefully we'll have time for questions and answers at the end. So the challenge of studying climate change impacts on any marine ecosystem, or any ecosystem for that matter, is a matter of several different scales. So it can be whether or not we're talking about looking at a single stressor, such as just looking at the impact of higher CO2, looking at two stressors, let's say CO2 and temperature, or trying to get to the more realistic realm where you have multiple stressors impacting your organisms and your ecosystem at the same time, which is more realistic. In this figure, these red triangles represent the proportion of studies that have been done. So this triangle tells us that most studies have been single stressor studies, and we're just moving into conducting multiple stressor studies. But again, that's really where we want to go because that's closest to the real world. If we look at the scale of the studies, we can see that the majority have been conducted on single species or strains of organisms, with less as we move up towards communities and ecosystems, and also less as we move to the finer scale of the molecular and cellular side. And then looking at the duration of these studies, the majority of the studies have been short-term studies conducted for months, hours, weeks, and months, very few on seasonal, annual, or decadal scales. Sorry about that. So we need to move from single species or single strain studies so that we can start understanding climate change impacts at community and ecosystem levels, but also try to get at the mechanisms which are at the molecular and cellular levels. And so another way of looking at it is we have these environmental perturbations, which are usually, or in nature, are always multiple stressors, such as temperature, CO2, and other. You get a physiological response, and what we hope to do is move from just looking at the physiological response to better understanding the population response, community response, ecosystem response, all the way up to understanding how all of these different levels affect global biogeochemical cycles. So this slide really lays out the grand challenges, and if you want to get more detail about this, this slide and a lot of this work is based on an NSF OA PI meeting that we had, and there's an oceanography manuscript that Andreas Anderson and myself led, where we go through some of these challenges and lay out a framework for future research. So if people are interested, you can get more detail about some of this introductory material in that paper. So I mentioned I talk about the experimental toolkit. So there's a variety of approaches that researchers all around the world are using to try to better understand how climate change is affecting organisms and ecosystems. The first approach, that's probably the most widely used approach, especially for ocean acidification work, are replicated aquarium studies. Then there are these aquarium systems where you can get at the mesocosm scale, so you can set up mini ecosystems that you replicate to try to get at population, community, and potentially ecosystem level impacts. Then there are groups that have taken these mesocosms and moved them in situ or into the natural environment. So these are, this is a photo of Ulf Rebussel's floating plankton mesocosms. So these things move around with the currents, and they're massive, and they can be replicated, and they have natural communities of plankton within them that are exposed to almost natural environmental conditions. Then there are short and long-term in situ manipulative experiments like the FOS studies that I will talk about. These are free ocean carbon enrichment studies that were originally developed by Ambari and that I've worked to modify for coral reef and shallow benthic ecosystems. Then there are also vent and seep studies where people have used natural ecosystems that have naturally high levels of CO2 to try to get a better understanding of what future ecosystems are going to look like and how they're going to change under high CO2. People have also done studies looking at natural gradients of pH and temperature and other environmental variables that occur naturally in nature, and people are doing transplant experiments and other studies to try to take advantage of these natural gradients to learn about organism and population and even ecosystem responses to climate change. There's also long-term sites, or the NSF LTER sites are a great example. These are sites that have been studied in many cases for multiple decades, and by trying to examine the changes to the ecosystems and the organisms over time, we can try to piece apart climate change impacts. And then there's also the approach of going back in time. So corals and other calcareous organisms have built skeletons that, when you look at isotopes and banding patterns, you can actually try to reconstruct past climate conditions, and you can also try to correlate those past climate conditions with changes in communities of organisms. So looking back over hundreds to thousands of years provides a pretty interesting window for trying to understand climate change impacts on different marine communities. And then to bring this all together, a very important component are numerical models that try to make sense of all these different types of data so that we can come up with mechanisms and models for how these different stressors are affecting key processes like photosynthesis and respiration and calcification, and how these might affect global cycles. So really the modeling and working closely with modelers is essential for integrating all these different types of data to really get a kind of a bigger picture and to be able to predict how changes that are predicted to occur in the future might affect ecosystems, populations, and communities. But each of these studies and approaches has several advantages, but they also have several disadvantages. So this is a table that, again, we put together with about 30 PIs in a room at the NSF OA meeting that was in Washington, D.C. a few years back. And I'm just going to highlight kind of the strengths, the key strengths and weaknesses of these different approaches. So I highlighted in green what these different approaches do particularly well. So you can see that aquarium studies, the first column here, they're excellent for replication. So you can get high levels of replication, often at a fairly affordable price. You can have very tight control over carbonate system parameters. They're an excellent system for studying multiple drivers, and I mentioned you can keep the cost down. They're good for studying both acute and chronic effects, and depending on the organism, they can be used to study adaptation and acclimation, especially when working with organisms with short generation times. So when we move to mesocosms, you can see that the natural realism improves a little bit. You can also have many of the same advantages that you have with aquarium studies in terms of the replication and control over the conditions. And sometimes you can study adaptations, sometimes you can't, depending on how these are set up. In situ mesocosms, they're much better for, much better for natural realism. They're a little more difficult to replicate because of the cost and logistical challenges of doing studies in the field, but they can give you good control over the carbonate parameters, and they can be good for acclimation studies. When we move to benthic FOS type studies, again, they're more realistic. They can be good for replication, but that is totally dependent on your budget. So typically, in my experience with FOS systems, you run them with a minimal number of replicates because of the time and cost involved. So let's say 3 to 6 replicate chambers in total. But if you had millions of dollars, there's no reason, and a huge team, there's no reason you couldn't up those numbers. So there's a possibility to have better replication. You do have good control over the carbonate system parameters. Bents and seeps, other natural gradients and long-term sites, again, they're best for studying natural realism. They're really good for looking at population and community effects, chronic effects, and indirect as well as direct effects. And some of the limitations of these studies, as I mentioned, aquarium studies, it's basically taking the organisms and putting them in a glass box. So often the flow conditions are different, the sea water conditions are often quite different, food can be quite different, light can be different. So that's really one of the big negatives of aquarium studies. It's very difficult to study population and community effects in a small glass aquarium. Mesocosms get around some of these changes, but it's very hard to run them long enough to look at processes such as adaptation. In situ mesocosms and FOS, you can see one of the biggest trade-offs is the cost, and with their high cost, it often means that the replication can't be as high as some of the other approaches. Bents and seeps, natural gradients, and long-term sites, you have no control over the carbonate chemistry, and there's often multiple drivers occurring at the same time, so it's very hard to piece out the impacts of any single driver, which makes it challenging. You also can't get at acute effects, and often, as I just mentioned, it's very hard to piece apart the direct effects of any of the different drivers. So you can see that any approach you use is going to have some advantages and disadvantages, and that's why I think it's so crucial to combine a variety of different approaches to really try to get a more well-rounded understanding of impacts. So today I'm going to talk about how I tried to kind of put this practice into effect by combining a variety of different approaches. So I'm going to briefly talk about some work I did on the Great Barrier Reef using a mesocosm study in a system that I developed there. I'm going to briefly talk about some aquarium studies that we did in parallel to the mesocosm work. I'm then going to talk about a 200-day in situ FOS, or free ocean carbon enrichment experiment, that you can see in these photos on the right of the screen. And then I'm also going to talk about a field study that we did that we compared present-day reef calcification and dissolution measurements with those that were taken using similar methods 40 years ago to see how rates of net calcification would change. So for all the work I'm going to approach today, it's really important to understand that when we think about coral reef growth, rather than just calcification, coral reef growth is actually a balance between calcification and erosion. So if we look at reef calcification, coral growth can be 10 to 300 millimeters a year. but reef accretion is only 1 to 3 millimeters per year. And what makes up for this difference is erosion and dissolution processes. And these photos on the bottom of the screen, these are various sources of erosion and dissolution. So this is a polychaete worm on the left. These are often internal bioeroders that can basically erode the coral skeleton away from the inside. There are external bioeroders like this sea urchin that can graze away the coral skeleton externally. There's physical erosion from things like wave energy, and then there's also chemical erosion or dissolution from changes in the carbonate chemistry that can lead to dissolution of skeletons. And so when we look at net reef calcification rates globally, we can see that many reefs globally are already close to net dissolution. So a value of 0 means there's no net calcification, that basically the calcification is exactly balanced by the dissolution and erosion. And we can see, especially in areas that typically have upwelling, we have regions that are shown with the red stars that already have negative net reef calcification, which means the overall balance is actually negative. So there's more dissolution than there is calcification, and over time these reefs will actually dissolve. But we can see it's not all terrible. There are reef areas that have positive net reef calcification, sometimes as high as plus 8.3. So this kind of gives you an idea that already there's quite a range of different net reef calcification rates, and we can learn a lot by studying why these rates are different in different parts of the world. So I mentioned I was going to talk about some aquarium and mesocosm studies. This is the Heron Island Climate Change Facility that I built on Heron Island when I was doing a postdoc there with Ovehue Goldberg and Sophie Dove. So you can see there are these 4 massive 10,000 liter sumps where we manipulate both the PCO2 and the temperature, and then this manipulated seawater is distributed to 72 replicate aquaria, which are all color-coded, and also to these 12 500-liter mesocosm tanks where we can produce miniature coral reef ecosystems that can be exposed to our 4 different environmental scenarios. And what was critical, I think, that we did in this work was, rather than doing our conditions as a flat line, we actually collected all our organisms from a reef location where there was a CSIRO buoy, where we could get real-time temperature and CO2 data, PCO2 data, and we did all of our treatments as an offset from what was being measured on the reef. So in this figure here, showing temperatures, this black line here is the actual temperatures that were measured on the reef, and the blue dots are the conditions that we produced in our control tank. So you can see that our control nicely mimics the variability you get in the environment. And then we had a pre-industrial treatment where we actually lowered the amount of CO2 and cooled the system by about 1 degree. And so you can see there's a nice degree offset below the control conditions. And then we had a B1, or kind of a middle of the road scenario, which had about 2 degree higher temperatures and about a 0.4 pH decrease. And then in red, you see we have the kind of business as usual scenario where the temperature increases by about 3 to 4 degrees Celsius, and the pH is dropped by about 0.5 pH units. And these black dotted lines actually show NOAA's bleaching thresholds that were predicted, and you can see that the control only reaches it right at the end of the study. The pre-industrial treatment never goes into bleaching conditions, whereas the middle of the road scenario reaches bleaching temperatures after only a couple weeks, whereas the business as usual reaches these bleaching conditions after about a week. So you can see that in a high CO2 world, we're likely to surpass coral's bleaching thresholds much more quickly, and we might have massive bleaching events much more commonly. As I mentioned, we also manipulated the CO2 levels in each of our treatments, and this is again to show that the blue are the controls which tried to mimic the CO2 levels measured on the reef. Again, the green, we lowered the CO2 levels, so the dissolved inorganic carbon actually went down, and you can see that it did this as an offset below the control conditions. And then again, you see the yellow middle of the road and the red business as usual scenario. And so we exposed these communities of organisms. We tried to, I actually surveyed the reef by the CSIRO buoy, and we tried to recreate the same coral coverage and diversity, as well as the same relative coverage of the dominant invertebrates in each of the 12 mesocosms. So you can see the 12 mesocosms have very similar corals. We actually also collected the sediments from the site where we collected the corals. You can see there's sea cucumbers. You can't see it very clearly, but there's actually several different fish and several different invertebrates that we tried to recreate the same amounts in each of them. And you can see each of the 12 tanks have these kind of blue-green filters. And these blue-green filters, they're actually made by a Hollywood lighting company, but they tend, we found ones that actually produce the same spectrum of light as the light where we collected the corals at about 5 meters. So we also tried to, as best as we could, replicate the light environment that the coral communities were exposed to. And I'm not going to show you a lot of the data. This study was published in PNAS in 2013. But basically, the big story is that when we looked at the changes in net calcification rate, one very interesting result is that the pre-industrial mesocosms had significantly higher net calcification rates than the controls, which suggests that if we went 200 years back in time, that coral reefs were likely growing faster and better than they are now. We also saw a significant decline between the controls and the middle of the road scenario, but most interestingly, when we went to the business as usual scenario, we saw a switch from net calcification to net dissolution. So we basically created one of those reef ecosystems where there was basically negative net reef calcification. So now I'm going to talk about some in situ OA coral experiments, basically taking these mesocosm and aquarium studies and putting them right on the reef. So this system, as I mentioned, we worked with the engineers at Mbari to develop the first faux system that they made for the deep ocean. They actually did it with only one replicate, again, because of logistical limitations. They basically chose the hardest place in the world to start doing a faux experiment, the very deep ocean. Their faux study could only be accessed with ROVs and AUVs. So as you can imagine, that's quite expensive and very difficult to do. We tried to take their system design and develop it for the shallow benthic ecosystems. And you can see, since this was a climate change experiment, we tried not to generate a lot of CO2 in the course of our experiments. So we actually built a wind generator that provided the majority of the power, as well as a solar panel that was on our float to provide most of the energy for our experiment. So this is an engineering diagram that explains how we controlled the pH actually quite accurately in situ. I'm not going to go into great detail. Basically, I'm just going to describe in a nutshell how it works. Basically, there's a series of custom National Instruments computers that would get the data from the array of sensors associated with each faux flume. So there was a Doppler velocity current, which would tell us the direction and speed of the flow within the flumes. I should mention the flumes were open at either end, so fish and other mobile invertebrates could come in from either end, and natural seawater regularly was flushing through either end. We had 1 to 3 pH sensors within each of these flumes. We also had a CDD to measure conductivity, temperature, depth. We had a PAR sensor so that we could get light readings. And the key sensors for the pH control were actually the pH sensors and the flow. So as I mentioned, the flow would tell us the direction and speed of the flow. So let's say the flow was running from north to south. That would tell the computers that if the pH measured was different than the desired offset, that it would then turn on the dosing pumps in the direction of the flow, adding CO2-enriched seawater until the desired offset was reached. And our system was a feedback system, so it was constantly taking measurements and turning on and off the pumps to get the right amount of CO2 addition to produce future predicted CO2 levels. So this is what the system looked like when it was deployed on the Reef. You can see there's 4 of these replicate chambers. These are the MBARI modified seabird pH sensors. You can see that some chambers have 1, others have 2, and even others have 3. I should mention that there was a suite of environmental sensors. So you can see the flow sensor here, the CDD, the pH sensor, and the light sensor. The reason environmental sensors were so key, one is it gave us a really excellent record of how the environmental conditions were changing throughout the experiment. But most importantly, like the mesocosm and aquarium studies, we did all our treatments as an offset from the pH measured in the environment. So it was very critical to have this real-time measurements of what was happening in the environment so we could do the treatments in our FOS flumes as an offset from what was occurring. You can see the wind generator that I briefly mentioned. Here's our control and logging system. I mentioned that MBARI chose one of the most difficult places to do a FOS experiment working in the deep sea. We tried to choose one of the most logistically feasible places, so we did it on the Reef flat, where at low tide we could actually walk out and service all of the instruments in the system. And even at high tide, the system was accessible by snorkel, so we didn't have to dive, which made everything a lot logistically simpler, which was quite important since we ran this experiment for 200 days. I should mention that we had 2 of the systems set as controls where we tried to replicate the environmental conditions, and then we also had 2 treatment flumes. So looking down inside each of the flumes in this study, we used Porites cylindrica, which is a branching Porites, which was the dominant coral on the Heron Island Reef flat. And in each flume, we put 5 colonies of healthy Porites cylindrica, and then we also had 5 Porites cylindrica in which we had removed the coral tissue to represent corals that had recently died from coral bleaching or disease. And I should mention that each of these 5 fragments were from a different mother colony to try to get the same genetic diversity within each of the flumes. So we ran this for 200 days, and when we were looking at the living corals, when we looked at their change in weight or buoyant weight, which is an estimate of net calcification rate, we could see that the control corals calcified about 8 to 9% per year, while the treatment had no net calcification. So there was a significant decline in calcification, in net calcification in the living corals. What was really interesting was when we looked at those recently dead corals, we could see that with the dead corals, there was about a 5% rate of erosion. But in the treatment, that rate doubled to more than 10%. So when you just look at the gray bars, you can see in the controls that if you take the net calcification minus the erosion from the dead corals, you can see roughly there's still net calcification if a reef was composed primarily of these Parietes cylindrical colonies. Under the high CO2 conditions, you can see with the calcification declining to no net calcification and a doubling in the rate of erosion and dissolution, that clearly under high CO2 on the reef, that you're going to shift towards net dissolution. And you can see that the results are more dramatic than what we saw in the mesocosm studies. And I think the reason for this is when you do an experiment in situ, the corals were exposed to the natural community of both internal and external bioerodiors, so the rates of bioerosion were likely much higher than they were in the mesocosm study. And so when we take all of our FO studies and we look over the 200 days at the change in the aragonite saturation state, you can see that in the live corals, which are shown in red, that they don't start to experience negative net calcification until an aragonite saturation state of 2.6, whereas the dead corals started to dissolve, have net dissolution at an aragonite saturation state of 3.8. So what this suggests to me is that maintaining living coral tissue is critical for protecting coral reefs, because an aragonite saturation state difference of 1.2 is pretty massive. So living corals do pretty well until the saturation state of 2.6 is reached, whereas the dead corals start to dissolve much more quickly. So basically, any reef management practice we can do that increases the amount of living coral tissue will increase the time we have until reefs potentially shift to net dissolution. So I think this in situ study actually provides some pretty important management considerations. Moving on from the FO study, I'm going to briefly talk about some reef biogeochemistry studies. The study I'm going to talk about now is a study that was done, that we did at One Tree Island with Jack Silverman and a series of collaborators from Israel. This study was led by Jack Silverman, and I was lucky enough to be part of it. One Tree Island is a reef system just next to Heron Island. It's a reef ecosystem that has been studied extensively for a long time. Donald Kinsey, who's one of the kind of godfathers of reef biogeochemistry, actually did his PhD and a lot of his early work there. And we actually chose several of Donald's sites that he studied 40 years ago and took net reef calcification measurements for the entire reef 40 years ago in the early 1970s. And we repeated those same measurements at the same sites, and what we found is over 40 years that the net reef calcification decreased 44%, and most interestingly, the reef dissolution at night increased 300%. So in this figure here of net calcification, G net or net calcification, you can see at night that there's net calcification, that the millimoles of calcium carbonate produced per day actually goes negative at night, and then you can see the calcification during the day, and this decline at night was 300% greater than it was 40 years ago. Part of this was likely due to changes in the reef composition, but it's quite likely that a lot of this change may be associated with changes in the chemistry that have occurred over the last 40 years. So I think if you take all these different studies together, it's clear that with declining coral calcification and increased bioerosion and dissolution, that reefs, many reefs, will likely dissolve in a high CO2 future. So this is a photographic scenario that Hugh Goldberg produced in a 2007 science paper, and you can see that reefs will likely change dramatically if we don't reduce our CO2 emissions, but it's not that all corals will be gone. Even in the worst scenario, there's still corals, but we'll probably lose some of the diversity, and the reefs will likely shift from net calcification to net dissolution. And I think we now can say this pretty confidently because we now have in situ data, mesocosm data, natural field studies, aquarium studies, and modeling studies that all produce these same conclusions. So when you integrate them all together, it does suggest, unfortunately, that if we don't reduce our CO2 emissions, that reefs will shift towards net dissolution. Briefly, I'm going to talk about some of the next steps I've been working on. So basically, these in situ studies, we've been trying to make portable systems so that we can compare reef ecosystems and actually different types of marine ecosystems in different areas. So we could set up a FOS study at, let's say, 10 reef sites globally and see which sites were most resilient, which were most sensitive to the changes. We could also compare different parts of the reef. Not all parts of the reef will respond the same way to ocean acidification. The Reef lab, where I conducted the FOS experiments, has large daily and seasonal swings in the environmental conditions, and it'll be interesting to see if the organisms in a highly variable environment are more or less sensitive to future CO2 increases than ones that have a more stable CO2 and temperature conditions. I'm also working on making these in situ studies multi-stressor, so we're trying to work out ways to do temperature, nutrients, and oxygen as well as CO2. And as you can see on the right, we're trying to modify our systems to deploy on intact communities and in different environments. And you see we're trying to go big. So this diver is actually to scale. So we're actually trying to enclose or partially enclose large natural ecosystems so that we can see how they deal with this different combination of stressors. And these new systems I've been working on with Christian Wild in the University of Bremen for a European Union consolidator grant. So we're hoping that the EU will be able to kick in a couple million euros to help us develop this kind of next generation FOS system. I'm also been working pretty hard on a new recently funded NSF that Martine Tresquers and myself wrote, and that we've been collaborating with Andreas Andersen on. And in this study, basically all I'm going to say, we just were in Panama for 6 weeks doing these experiments, so I can't talk about the data yet, but we're trying to combine aquarium studies, in situ transplant experiments. So you can see these are large transplant studies with 3 different species of corals, with cellular physiology methods that Martine Tresquers and his lab have developed, with large reef biogeochemistry studies that Andreas Andersen is leading and has done a lot of work with, so that we can try to get at coral calcification mechanisms so we can understand on a cellular level how changes in CO2, temperature, nutrients, and other stressors affect internal pH regulation. And I think it's really critical that we move from understanding organismal, population, and community impacts to actually trying to understand what's happening on the cellular and molecular levels, because if we can understand the mechanism, we're much more likely going to be able to develop some sort of solutions to try to protect coral reefs in light of climate change. And I should mention the work I presented today has been done with a massive team of collaborators. I'm not going to go through all of them, but it includes collaborators at the University of Queensland Coral Reef Ecosystem Lab, where I did a 5-year postdoc with Sophie Dove and O.Q. Goldberg. The FOS system was developed with the UQ engineering program that was led by Malcolm Marker. As I mentioned, it was also a collaboration with Ambari and their team of engineers in Peter Brewer's group. We work closely with Rob Dunbar's group at Stanford, as well as the physical oceanography group, Jeffrey Kosep and Stephen Mono-Smith at Stanford, the Ken Caldera Lab at Carnegie, Brad Opdyke group at the Australian National University, Malcolm McCulloch's group at the University of Western Australia. We also work closely with on these reef biogeochemistry studies with Jack Silverman, Tanya Rivlin, Boaz Lavar, Jonathan Ayres, and Kenny Schneider at the Hebrew University. And as I mentioned, this NSF Cellular Physiology project is done in collaboration with Martine Trescares and Andreas Anderson. And this work has been funded by the National Science Foundation, the Australian Research Council, and the Pacific Blue Foundation. And with that, hopefully we still have some time for questions. Thanks a lot.
Sure, David, thank you so much. That was fascinating. So we do have a question, and it is from Jenny Vander Fluem. Excuse me, Jenny, if I messed your last name up or did not pronounce it correctly. And she wants to know, is there any interest in including non-coral hard-bottom reefs of limestone base similar to those found in the off the southeast U.S.?
Definitely. I mean, I tried to design the faux systems so that they could be deployed in any shallow benthic environment. I'm working on making them deployable up to 30 meters, and just by minor modifications to the chamber design, we should be able to deploy on a variety of different environments. So the type of environment you're mentioning, we should definitely be able to do experiments on. It just comes down to getting the funding to do the studies and build the systems.
Okay, great, thank you. And then following up on that, I guess getting a little more specific, there's a question, how do you think the faux systems will work in a shallow water estuarine environment, for example, over oyster beds?
Again, I think it would be an ideal location to deploy the faux. I wouldn't foresee any problems, and I think doing it over oyster beds would be very interesting because you could look at changes in the oyster calcification, and you could even look at things like the oyster disease under high CO2, which could be quite interesting and quite relevant for the shellfish industries.
Right, exactly. Okay, another question from Astrid Schnetzler. She wants to know how much does your faux approach allow for the consideration of plankton assemblages and other pelagic organisms?
Right. So a critical aspect of our faux design so far was to leave either end totally open so that natural planktonic communities could flow through. So with corals, for instance, there's a lot of papers coming out that show that the food levels are really critical to the coral's response. So when corals have natural plankton communities that they can feed on, they're often more resistant to changes in CO2 levels. And in an aquarium system or mesocosm system, it's very difficult to recreate that natural planktonic community. The faux, you have that natural planktonic community, and I think that's one of the critical advantages of using the faux system. So I would say this is actually one of the key strengths of the faux system, is you do get that natural planktonic community, which for corals is really critical for their food and their kind of overall energy budget.
Okay, great, thank you. And then let's see, the question is, what is the cost to build a shallow water faux experimental system?
Sure, so the cost depends on the number of replicates, the size of the flumes, so the area you want to enclose. I would like to say I could do it really cheap, but the problem is if we're doing these studies in shallow benthic ecosystems, you really need quality instruments. So, I mean, for example, a sea fox that measures pH and oxygen that was developed by Todd Martz and others, that can be $20,000 to $30,000 for one sensor system. So if you want to have a system with 4 to 6 replicates, you can see that if the pH sensors, the flow sensors, which can also be $10,000 to $15,000, CTDs, that we're quickly talking about $200,000 to $300,000 just for the instrumentation. And besides that, you need the custom control systems, the power systems, and the communication systems. So not to scare everyone off, but I think probably for a minimal faux system like the one I did in Australia, you could do it for about $500,000. A more advanced system, that's larger scale. The one we're trying to develop for this EU grant, just for the FOS development, we put in $750,000 in addition to the salaries of everyone working on it. So it's not cheap, but it's also, I think, for the amount of information and data you can get, it's not totally unreasonable either.
Okay, great, thank you. Stephen Jameson asks, how do you extrapolate impacts and conditions at the cellular level to impacts and conditions at the ecosystem level?
Oh, that's a great question. Again, it's by trying to study kind of the impacts on the multiple levels at the same time. So, like with our transplant experiments, we try to characterize not only the biogeochemistry, but the all of the environmental conditions, so that we can put the cellular physiological changes into as deep ecological context as possible. And as I mentioned before, working with modelers is really critical. So basically, we need to integrate this cellular physiology data with all the biogeochemistry and ecological data that we have to try to do what you asked, which is not an easy thing to do, but it is our holy grail. That's what we're trying to do. And I would say, hopefully I'll have a better answer to that in the next year or so, because that's actually what we're trying to do right now with the results that we just collected in Bocas del Toro, Panama.
Okay, great, thank you. And Astrid also wants to know, how realistic is the notion that we will end up with soft coral reefs globally?
So I'm not sure I totally understand the question. Is it that hard corals are going to disappear and we'll just have soft corals left?
Astrid, if you're there, if you want to clarify, I'm just reading what she typed. How realistic is that we will end up with soft coral reefs globally? I guess that's what I would surmise, is that she's...
Okay.
Yes, she is, yeah.
I would say no, simply because I think there still will be hard coral reef ecosystems. They're just going to look very different than what they do today. So from those volcanic seeps and vents, we know that corals like Porites, massive Porites, are kind of like the cockroaches of the corals, and they survive. So we'll probably still have some hard coral species. There just won't be as many of them, and they'll be dominated by these weedy kind of cockroach species like Porites. That's what I would predict.
Okay, thank you. All right, if there are any other questions, if you could please type them into the question box. And I don't see any coming in right now. So what I would like to do is I'll add that Jenny added that she thought your presentation was excellent. So thank you.
Thanks a lot.
Yeah, sure. And again, thank you. It was a fabulous presentation. So if any of you as participants think you have questions later, you're welcome to contact David via the email. His email, which you can find in the right-hand side of the control panel in the chat box. And if you are interested in joining the SOCAN listserv to learn about future updates in the Acidification Network, please sign up at the URL shown on this slide. And please direct any questions about the SOCAN network to socan@secoora.org. So again, Davey, thank you for a fabulous presentation. And we'd like to thank all of the participants for attending the SOCAN State of the Science webinar this afternoon. We welcome any feedback you have or any further questions or suggestions for topics for this webinar series. And you can submit input by replying to the following, by following up to the email that you will be receiving from us shortly. The recording of this webinar and a PDF of the presentation will be available on the SOCAN website with the URL listed here. Once again, if you'd like to stay up to date on SOCAN happenings, please do sign up on the SOCAN listserv. Thanks again, Davey, for your very informative presentation, and thanks to all of you for your thoughtful questions and taking the time to join us this afternoon. This is the end of today's session.
Thank you. Bye.
Thank you. Bye-bye.